Hello everyone, my name is Rafael Zas and I'm a co-author of this collaborative study carried out by researchers from the Swedish University of Agricultural Science in Uppsala, Sweden, and from the National Research Council in Pontevedra, Spain. The study, published in Forest Ecology and Management, explores whether we can protect conifer seedlings against forest pests by improving their defensive status through the application of methyl jasmonate, a phytohormone involved in the recognition, signaling, and triggering of induced resistance in plants. Particularly, we explored whether methyl jasmonate can be used to protect seedlings against the pine weevil Hylobius abietis, a chewing herbivore that feeds on the bark and phloem of conifer seedlings. This is an extremely problematic forest pest that causes extensive mortality and growth losses in juvenile seedlings established in clear cuts all around Europe. It is considered a major silvicultural pest for which no definitive protection methods are available. So new environmentally friendly and cost-effective methods are clearly needed to protect seedlings against this harmful pest. It is well known that conifer seedlings defend against insect herbivores by means of a battery of chemical and physical defenses that can be either constitutive, which are permanently expressed in the plant, or induced after the recognition of damage. In many situations, induced defenses can be vital for the plant to survive the attack. We now know that the jasmonic acid plays a central role in the complex molecular mechanisms that are involved in the recognition, signaling, and triggering of induced defenses. Accordingly, the application of this phytohormone to conifer seedlings elicits response similar to those induced by real herbivory, including increased effective resistance against several pathogens and pests. But all these previous results have been obtained under controlled conditions in the lab by means of in vivo or in vitro biocides. Whether methyl jasmonate can be effective to protect seedlings against the pine weevil under real field conditions remain to be tested. And this is exactly what we have done in this study, aiming to bridge the gap between success in the lab and success in the field. So our main question was to explore whether we can improve seedling resistance against the pine weevil by eliciting methyl jasmonate-inducible responses in pine seedlings at the nursery just before planting. To this end, we established two field trials in clear cuts, one in Sweden and one in Spain, with the four more commonly planted conifers species in both northern and southern Europe. The two trials were naturally infested by the pine weevil. We treated the seedlings spraying methyl jasmonate at the nursery a few weeks before planting, and we explored the effect of the concentration, number, and timing of application of methyl jasmonate on plant chemical defense, weevil damage, and plant growth over two growing periods after planting. The results were clear and conclusive. As we can see in this figure, methyl jasmonate strongly increased resin-based chemical defense in the four species, and the effects were generally proportional to the concentration used. The increase in chemical defenses was translated into reduced damage by the pine weevil once the seedlings were established at field. As shown in the two top figures, the application of methyl jasmonate reduced the debarked area caused by the pine weevil in a dose-dependent manner. And this reduction of damage was biologically significant, as it was translated into a significant reduction of seedling mortality, as shown in the two bottom figures. The application of methyl jasmonate was especially effective on Scots pine seedlings, where the treatment decreased the mortality from 40% to 7% after two field seasons. So we can conclude that methyl jasmonate was effective protecting conifer seedlings against the pine weevil at real field conditions. The effects were mostly dose-dependent, and the best results were obtained with repeated application of methyl jasmonate. So in summary, the results of this comprehensive field study give enough evidence to affirm that methyl jasmonate has the potential to become an attractive, environmentally friendly, and cost-effective alternative way to fight against the pine weevil.